So now we're going to start our official morning session, talking about different states and provinces and tribal efforts with test and remove programs. And our first speaker for the morning, we're going right back to Francis. So I think, Francis, if you want to go ahead and share your screen, we'll go ahead and have you take it away. Okay, we can see it.
Guess it would help if I went off mute. Sorry. Thank you. Okay, so I'll, this talk will be about our experience with continuing on after a SOTN. Okay, so in Hells Canyon, most of the populations are interconnected, as we found by the spread of MOV, but they're not all connected to each other. And this is just a little background of the setup for this, what I'm going to talk about next. And the sizes of the individual populations are really small to over 100. And the accessibility is, very accessible to pretty much inaccessible or not very accessible. The populations have been stable to declining, and our objective is to increase them. So when we first started, our initial plan was to have treatments and controls. And these, as I mentioned earlier, that the populations were selected by their accessibility, and most of them were pretty small. So we were going to conduct treatments in 3 populations and have 2 populations as controls. The test and remove project, how it actually ended up is, of course, how this goes when you try to do field work. So we started out with our treatment in a SOTN and the controls. And then the next year, first of all, we had a new strain that was introduced at Heller Bar in the Black Butte population. And we got really concerned about that and decided that we were just there were only 8 ewes there. And we thought they were pretty well isolated from other sheep. So we removed all 8 sheep. Turns out the strain still spread elsewhere. But anyway, we removed those sheep and brought them to our captive study. And then we also had a population down the south part of the canyon called Sheep Mountain. And it was just 3 ewes. And I think as far as we knew, there were no rams. And we kind of wanted them there. The managers kind of wanted them there as a placeholder so that the federal land management agencies would still regard this occupied sheep habitat. But there was some concern, particularly by Oregon, that they were going to infect the Lookout Mountain herd, which at that time was healthy. So we removed those 2 and brought them to captivity. And then we did end up removing ewes from the Lostine, so that became a treatment population later on. And then because of as I mentioned, because of our encouraging results from a SOTN, we decided to go ahead and start to see what we could do in the rest of the canyon. And so we pretty much have tested and done intensive testing in 6 populations and tested all the other populations, except for 2 that are really hard to get to, and there's not very many sheep. Upper Hells Canyon, Bear Creek, and Bear Creek, there are actually a good number of sheep. We do have limited testing from Bear Creek, and it seems to be negative, but we haven't found a way to logistically test them. Okay, so next few slides are going to be timelines of the MOV PCR status of different populations. And in 2014, we tested, as I said, we moved into some of the populations elsewhere in Hell's Canyon. And that was also the last year of the Asotin study. And kind of, one happened after the other, but they're still in the same year. And we found 10 adults that were positive, and 9 of those were removed. We removed the 3 Sheep Mountain sheep and the Black Butte sheep, of which 3 of them were carriers, 8 were positive, and 5 of them were not. And we also removed 2 sheep from the Lostine. And then we removed 3 sheep, the 3 sheep from Asotin that I mentioned earlier. And then the 1 positive sheep that we had in Mountain View died. So after this year, we never detected any MOV. Well, we had no more sheep at Sheep Mountain. We never detected MOV in any of the other populations where we removed the sheep. And then in 2016, we went into some other populations. We did some retesting and went to some other populations, and we found 12 sheep that were positive. 1 positive adult died, and the rest were negative on retest. Whoops. So basically in these populations here, one of the sheep that were positive, one of them, actually 2 of them were sheep that were positive. 1 died, and then 2 of them in different herds, when we went back to retest them, they were negative. Then the next year we had 2 rams in Redbird and 2 ewes in Lower Hells Canyon that were positive. And both of the ewes, and we were going to retest the ewes in both of the ewes in the Lower Hells Canyon, sorry. And but both of them died before we could retest them. And the rams, consistent with our hypothesis, we just left in the population. And we did remove 6 sheep from the Lostine that were carriers. And so the last, at the end here, we went back and retested populations over the next 2 years, and we haven't detected any MOV or pneumonia, and except for in the Lostine, where we're still seeing MOV, and we removed 1 carrier. So a summary of our work in Hell's Canyon is that we sampled 11 populations, not including the Lostine. So all this data does not include the Lostine. There were a total of 605 sheep in those 11 populations. We tested 396 of them, which is 65%. There were 175 ewes in our 6 intensively sampled populations, and we sampled 94% of them. The other populations that were not intensively sampled are populations that we tested and then found out that MOV had cleared naturally. So we didn't, based on our criteria of sampling lambs and ewes. In terms of the results, we found that we found 43 MOV positive sheep for all ages of sheep. So 11% of the sheep we sampled. And of the adults that we sampled, 10% were positive. We had 21 sheep. Half of those sheep either died naturally or they were negative on retest. And then we removed 9 of them. So in the whole study, we removed 9 sheep out of 396, 2% of the sheep that we tested. And then we also, we also removed other sheep. that were for our research. So with that, we had 10 populations that were previously MOV positive that now we are not able to detect MOV. And we have, unlike in DeSoto, we have seen some significant population responses. This is the redbird herd in Idaho, the Mountain View and Winaha herd in Washington and Oregon. And then of course, this was the Soton herd. And it's kind of amazing that, very coincidental that right when we detected the last MOV detection, following that was a significant inflection point in the population, except for poor Soton. So overall, out of the 10 populations, half of them have responded really well. They're increasing, and then 4 are not. They're kind of stable, and most of those, other than Soton, well, Soton's pretty small. It's got 20 to 30 ewes, like I said, and the other ones are even smaller. And so they're having the lambs are surviving, but the populations are not really increasing. They're just kind of maintaining. So my takeaways from this is why this happened is that I think Bighorn sheep immune strategy, such that it is to resist infection. Now, in the adapted host domestic sheep, the strategy is mostly to tolerate it. And we know that, and evidence that supports this is that domestic sheep flocks generally have a higher prevalence of infection than wild sheep. So more animals are just living with MOV, and they actually have a weaker immune response based on our ELISA test, and there's less disease. So therefore, I think that removal of the carriers and preventing re-exposure is assisting a natural process of clearance that is consistent with the immune strategy of the Bighorn sheep, along with the attrition, the natural attrition of carriers. I also think that large populations almost always have some substructure that can be exploited to effectively accomplish test and remove, or perhaps other disease management strategies. So I think it may be more doable to conduct test and remove in large populations than I had originally thought it would be. And there is variability within it across populations and situations. So this is Hell's Canyon. There's going to be, other people have seen similar things in some places and different things in others. And for example, even within Hell's Canyon, the lostine differs from the other populations. So in Hell's Canyon, our future direction is to focus on the lost, clearing MOV from lostine and now lookout, which you'll hear about today. And really trying to reduce the risk of new spillovers. And we have a domestic sheep and goat health testing program for that, to try to help with that. And I'd like to continue to monitor health less intensively and population trend, particularly for these last few populations that cleared. We only have a few years of data, and I'd like to be sure that they are actually cleared. And expanding test and remove it to other populations, specifically the tri-state, Idaho, Lower Salmon, Oregon Burt River, and Washington Yakima. Thanks.
Thank you, Frances. Excellent, and thanks everyone so far for staying on time. Really appreciate that. It's giving us lots of opportunity for questions. Okay, our next speaker From South Dakota is going to be Chad Lehman, who's a senior wildlife biologist with South Dakota Department of Game, Fish, and Parks. And he works extensively with all game species, but I bet he spends the majority of his time working with sheep, and especially in Custer State Park. So welcome, Chad. And if you want to go ahead and share your screen, we'll get started. Gotcha.
Right. Okay. Well, good morning, everyone. Thanks again for the introduction, Perry, and I look forward to sharing some information here in South Dakota. I need to acknowledge some other co-authors on this. Jonathan Jenks, retired professor from South Dakota State University, has done a lot of work in this field. He wasn't able to join us today. And then I'm going to be tag teaming with MS candidate Amanda Ensrud at South Dakota State University, and she's going to go over some of her research as well. So I'm going to give a quick little background on South Dakota test and remove here. You heard Kevin Hurley mention 1922, and I think he may have been referring to Senator from South Dakota who reintroduced bighorn sheep into Custer State Park from Canada back in 1922. And since that time, we've seen precipitous declines in bighorn sheep populations throughout South Dakota. And then more recently, we had a pneumonia die-off in 2004, where we had 90% of our sheep population die. Rapid City herd in 2009, we saw 50 to 60% die off. And then in Deadwood in 2016, we also saw a major die off in that herd as well. I'm not going to go into the chronic carrier hypothesis. Francis did a good job of providing some background on that for everyone. And I'm just going to share the first case study here in Custer State Park. Tyler Garwood was our master's student on that project and did a good job helping us meet the objectives of that study. And our primary hypothesis was to take what Francis and Tom are discussing with the experiments they're doing and then applying that into a field setting in South Dakota. And so what we had was a treatment population in Custer State Park, and comparing that with our Rapid City control population. And our objectives were to determine if removing chronically carrying individuals would reduce the prevalence of mebile shedding, and ascertain whether chronic carrier removal reduced pneumonia-related mortality. So this just gives you a spatial depiction of where our two populations were located. We have the Rapid City herd, which at that time was about 70 bighorn sheep in 2016. And that was our control population that we left the chronic carriers within that population. And then in 2014, when we first started testing sheep in Custer State Park, that was our treatment population. And those are the animals where we went in and tested and then removed the chronically carrying sheep from that herd. So materials and methods, we used VHF collars for both the ewes and the lambs and the rams that we were captured. We used chemical immobilization, aerial net gunning to capture and test all of the animals. We used vaginal implant transmitters in pregnant ewes to facilitate capture of lambs. And I probably will save some of I won't go into all the exact methods for all the testing. It's been pretty universal for a lot of these studies. So we started testing in August of 2014, and we tried to get at least 2 tests on each individual. So that included rams, lambs, yearlings, and adults for the entire population in Custer State Park. And we identified animals as chronic carriers, always testing positive, intermittent carriers, where we had both mixed negative and positive tests, and then non-carriers, which are all negative tests. So we had in Custer State Park, We identified 2 chronic carriers that always tested positive. Those animals were removed from Custer State Park and then were brought to South Dakota State University. And then when we did then is we then continued to test this herd post removal. And so for results then, after removal of the chronic carriers, no MOB was detected from 35 samples from 26 individuals. And then between January 2016 and May 2018, we collected 80 samples from the 57 control population Bighorn sheep. MOV was detected in 38 samples, or 48%. 9 samples are indeterminate and 32 are negative. And for our total sample size, then we had 86 radio-collared adults. And so for pneumonia mortality hazard for the treatment population, adults were significantly reduced relative to the control. Pneumonia caused less adult mortality in the treatment population than the control herd. And we did see a spike in predation in our treatment population in Custer State Park. We had a fairly high mountain lion predation rate. But when you look at actual pneumonia-related deaths, we did not document any pneumonia-related deaths subsequent then to the removal of the shedders. And when you look at lambs, we had a sample size of 43. Chronic carrier removal had a negative effect on the daily lamb hazard, which means we had a significant increase in lamb survival in the treatment population. We had a 77% annual survival rate in the treatment population compared to 35% within the control Rapid City herd. Pneumonia-caused mortality was significantly less likely in the treatment population than in the control population. So then moving into the Deadwood case study, this is a more recent study where we had 24 sheep remaining. We had noticed through our lamb to ewe ratios that lamb recruitment had been rebounding. It was 31% in 2020 versus 6% in 18. We started testing individuals. We tested 18 of 24. And so we tested the majority of the individuals without testing 1 ewe, 3 lambs, and 2 rams. And in 2021, all PCR MOV tests were negative. Additionally, we did we tested C. ELISA detection for exposure to MOV, and we had 1 seropositive detection. So what we suspect is occurring here is this could be an example of natural clearance of MOV leading to recovery. And so we're going to continue to monitor this herd, but we did not remove any carriers from this herd, unlike the Custer State Park or the Rapid City herd. And I'm going to let Amanda chime in here with the Rapid City case study. Amanda, are you there?
Yep, I'm here. At the start of the study, the Rapid City Bighorn sheep herd had 22 ewes and 12 rams, and we ended with 13 ewes and 10 rams, and that's only counting the yearlings and adults. There are actually 2 subherds that make up the Rapid City herd, so that's Rapid Creek and Spring Creek. They have the potential to commingle, but very rarely do. The Rapid City herd received the test and removal treatment from 2018 to 2020, and historical Rapid City herd data was used as a pre-treatment comparison. We've removed a total of 9 chronic carriers that includes 8 ewes and 1 ram, 2 of which were indeterminate or intermittently carrying ewes that were removed November of 2020. This was done for precautionary purposes, ensuring that no carriers of any kind persisted in the herd. These specific individuals consistently tested indeterminate with MOV PCR and regularly interacted with naive lambs. All known carriers have been removed, and each individual in the herd has been tested at least twice. During the course of the study, we had 66 nasal mucosal swabs that were tested with MOV PCR. That resulted in 46 negatives, 11 positives, 9 indeterminates. Next slide, Chad. Okay, So for, just going to go over the adult results a little bit. So at the top there are my two competing top survival models based on WAIC ranking. So the covariates that we used were beta test, which was a binary covariate, and individuals were given a 1 if they had ever tested positive for a MOVI, and a 0 if they had only ever tested negative or indeterminate for a MOVI. This was all based off of MOVI PCR data. And beta treatment was also a binary variable and varied by time and weeks, taking into account the individuals that were alive before and after treatment. So we had two competing top models here. And the first model had log unit cumulative hazard rate, beta test, and the weak effect with a model weight of 0.524. The second competing model was log unit cumulative hazard rate, beta test, beta treatment, and the weak effect with a model weight of 0.463. Analysis indicated that the chronic carrier removal treatment had a negative effect on the daily log cumulative hazard rate of adults with a beta treatment value of negative 1.64, although the probability of adults survival declined 1% from 88 pre-treatment to 87 post-treatment. This rather static adult survival was most likely due to the inclusion of chronic carrier removal as a source of mortality in the post-treatment survival. Chronic carrier removal was included in the mortality category human-caused in the following graphics. The probability of a pneumonia-related mortality in adults did decrease from 55% pre-treatment to 2% post-treatment. So no pneumonia-related adult mortalities were documented after the start of the treatment. The 2% mortality is only due to uncertainty in some mortality events, which was included in parameter estimates. Next slide. Okay, so here's the juicy stuff. So the most supported lamb survival model included the baseline log unit cumulative hazard rate, beta treatment, and the the day effect with a model weight of 0.802. Analysis indicated that the chronic carrier removal treatment had a negative effect on the daily log cumulative hazard rate of lambs with a beta treatment value of negative 1.23, which corresponded with an increase in lamb survival from 21% pre-treatment to 62% post-treatment. As illustrated in the figures, the probability of a pneumonia-related mortality decreased in lambs from 48% pre-treatment to 6% post-treatment. And again, there was no pneumonia-related mortalities that were documented after the start of treatment, and then the mortality, the percent for pneumonia-related mortality was just based off of the uncertainty. So we tested 2 of the 2020 lambs from the Rapid Creek subherd, and that was done this January, and they tested positive for a MOVI, none of which developed symptomatic pneumonia. This was after the removal of the indeterminate or intermittent carrier that had close contact with, that they had close contact with, and additional testing done this fall of 2021 of lambs will conclude if this was in fact the issue, or if there's an unknown domestic flock concern or another unknown threat. The Spring Creek subherd had, which the other subherd had all recruited lambs test negative this January for MOVI. So all in all, we're feeling very positive about the Rapid CD herd with great recruitment from 2020. So that's 8 out of 10 lambs that were born. And like I previously said, no pneumonia-related mortalities, and already have reports of seemingly healthy 2021 lambs. That's it for me. Thank you.
Okay, so to summarize this then, through our 3 case studies that we found pneumonia can be maintained in free-ranging bighorn sheep by a few chronically carrying MOVI individuals. But once these individuals are removed, we found where there were no deaths attributed to pneumonia following that treatment. We did have one case where we believe we have natural loss of the carriers, which I believe Frances alluded to earlier, that she saw that as well. So we did find that adult survival in the CSP study did average 10% higher than the control population. And then what Mandy found was she really didn't see a difference in adult survival before and after treatment. And she alluded to the fact why that might be occurring. However, lamb survival was significantly higher in both treatment populations. So given the cost of doing this work, it's very expensive to, radio mark and test all of the individuals within these herds. Managers might be wondering, why not just, versus testing all these animals, just completely remove the entire herd and start over. Well, one of the big things that we saw in Custer State Park is some of these animals have a lot of knowledge about, the area they live in, following green up, avoiding predation. They know where the migration corridors are. So I think there's some benefit in maintaining these older animals within these herds. And just an anecdotal side note, when we brought in Badlands sheep to supplement the Custer State Park herd, once we were cleared of it, it was interesting. We compared lamb survival between the naive ewes with the Custer State Park ewes, and there was a definite advantage for the Custer State Park ewes. They had much higher lamb survival the first year subsequent to the reintroduction of the Badlands ewes, where they only produced one lamb out of the 12 ewes that we brought in. Since that time, they have managed to produce more lambs, but it is interesting to note that I think it's important, if we can, to keep some of these older ewes with that population level knowledge in the herd. And then one last side note, in all of our populations in the Black Hills, we have very good road access. So our animals are definitely available to us to do a lot of this testing from roads. So I think that's something that has definitely benefited us doing this research and management as well. And with that, I guess we'll open it up for any questions.
Thank you, Chad. That was really informative. Actually, we're going to hold all questions till the end. So thank you for getting us ahead of time. And I think we'll go ahead and move on to our next presenter in this morning series. So I'd like to, and if you can, thank you for stop sharing your screen. Jack Hogg is going to be our next presenter, talking from Montana, and he is a Montana Conservation and Science Institute. That's his primary employer, which is a research and education nonprofit headquartered in Missoula, Montana. He's also a faculty affiliate with the Division of Biological Sciences at the University of Montana, and his primary study sites are the National Bison Range in Montana and the Sheep River Provincial Park in Alberta. And I believe today he's going to be sharing his experiences with the National Bison Range.
No, I can't get.
Jack, we could, if we wanted to go to the next presenter, you could email me your PowerPoint and I can share it.
Yeah, I had it going before earlier, but yeah, go ahead.
Okay, so email me your
Okay.
PowerPoint and we'll circle back to you.
Todd, are you out there?
Yes.
All right. Okay. So we're going to move on to Nebraska with Todd Nordeen, who's the Big Game Disease and Research Manager for the Nebraska Game and Parks Commission. And Todd, go ahead and share your screen and we'll take it away. Slideshow, next one over.
Yep, I'm trying to get that started, or whoops. There we go.
Yeah.
There, you got it?
It's should be.
You see it?
Yep, you're good.
Yes, now we do. Perfect.
All right. Yeah, good morning, everybody. I'm actually going to be sharing this presentation with Emily Moberg. She's our graduate student, also at South Dakota State University, working with John Jenks up there. But To start with, I'm just going to quickly run through the history of bighorn sheep in Nebraska, and then turn it over to Emily, who has just received a lot of the details, and she'll be sharing that with you when we get to that point. So you've all seen this distribution map over the years of what it looked like historically. And as you zoom into Nebraska, you can see what our historic range looked like, mostly covering the western fourth of the state. And they were extirpated, from the state by the early 1900s. And then we began reintroductions clear back in 1981, and then followed up with more in 2001, 2005, 2007, and 2012. Which has led us to what we have today for bighorn sheep back in Nebraska, with 3 distinct herds up in the northern region, which we classify as our Pine Ridge herds up in there and up in that area. And then down in the south, what we call our Wildcat Hills region, we have 2 herds down there. But our populations have certainly suffered, and you take a look at this screen here, our Pine Ridge herds are really declined. They're below 50 now. Our Sow-Belly, Fort Robb, and Barrel Butte herds each have about 15 sheep, or actually probably less than that in some cases. But on the other hand, our Wildcat Hills herd, our numbers are pretty good down there. Our Cedar Canyon herd on the lower left is currently at about 60 sheep, while our Hubbard's Gap herd in the lower right is currently at about 240. As far as disease events, the Pine Ridge herds up north have been hit really hard. All 3 herds have suffered significant events, and more than one actually. And we've seen that recurring low recruitment in all of those herds, obviously leading to those low population numbers that you just saw. And with the Wildcat Hills, as I kind of mentioned, we're seeing a different reaction down there. We did have a disease event in that Cedar Canyon herd in 2006, and followed by low recruitment for about 10 years, but we're now seeing that herd recover, and we've actually reached or matched our highest numbers ever with that herd. And our Hubbard's Gap herd, that's remained fairly clean for the most part, giving us those high numbers of 240 over there, which has ultimately led us to the research we're talking about here today, looking at the disease events here in Nebraska and testing this chronic shedder and test and remove hypothesis. So, and we've ultimately lined up Emily Moberg and John Jinks to help us with that. And so I'm going to turn it over to Emily now, since she has more of the details and the actual research. You there, Emily?
Yeah, I'm here. So South Dakota State University kind of took over this study in 2018, and we continued through early fall of 2020. And since then, I've been back at Brookings, kind of working on the analysis and trying to wrap up this project. But to break down our study area, we have our treatment herd, which Todd was talking about in the Pine Ridge, and then our control herd in the south, which is the Wildcat Hills. And then just to go a little bit more in depth, South Dakota State has only been really focusing on 3 of these subherds for our analysis. And those are highlighted on the left right there. So you can see Fort Robb is right in the middle of our Pine Ridge. Barrel Butte is on that east side. And then Hubbard's Gap is on the east side of our control herd in the south. So we are only really looking at those 3 and kind of are lacking a little bit of information more in depthly on the Sow-Belly and that Cedar Canyon herd. Since, I guess, February of 2017, Nebraska Game and Parks has been conducting annual ewe captures. In 2017, they only caught ewes in the north, in the Pine Ridge. But then in 2018, we expanded to Hubbard's Gap. So you can kind of see here how many we were capturing in the total population. We were aiming to catch every individual in the Pine Ridge, but with costs and some elusive sheep, we couldn't quite catch them all. And then in the Hubbard's Gap, there were many more ewes, so we only try to target around 20 to 30 each year. But by 2020, we were able to capture every single individual in the Pine Ridge. So since we started in 2017, we wanted 2 consecutive years of testing to remove any chronic shutters. So we classified a chronic shutter as any individual that tests positive for MOVI in those 2 consecutive years. If they had, a negative or indeterminate result, we still just classified them maybe as an intermittent shutter. So in 2018, we didn't do any removals. 2019, we removed 9 from the Pine Ridge. And then in 2020, we still had 1 last chronic shutter, or chronic carrier. So we removed her from the Fort Robb, and there were no more in Barrel Butte. So this sheet kind of explains the prevalence of MOVI in our herds. As you can see, in 2017, it was pretty high. So as we continued with our testing, it still remained high in 2018, but in 2019, we were successful in removing a MOVI from our Barrel Butte herd. Fort Robb, we still had a few maybe intermittent shutters, or that 1 lingering chronic shutter that we removed in 2020. The only tests we saw in Hubbard's Gap, we had 3 out of 20 that tested positive for a MOVI in 2018, but then after that, they were completely clear. And our tree over here kind of shows the different strains that you can see in each herd. And so that Hubbard's Gap herd is very distinct when they did have it. But otherwise, Sow-Belly, Barrel Butte, and Fort Robinson, you can see tend to commingle. So our adult female mortalities, in all, we had 52 individuals that we monitored in the Wildcat Hills. There were 9 mortalities, and we just kind of break down the different causes of mortality there. And then in the Pine Ridge, we had 31 individuals with 14 mortalities in the end. Predation was pretty high in the Pine Ridge. And I just did want to stress that our survival and cause-specific mortality analysis does not include any euthanized or removed sheep. So for our adult survival, I'm just reporting for this presentation our global hazard model, just because it includes all of our covariates, and I kind of want to just show how everything breaks down. So our beta treatment was, or I guess I'll go over our effects. Our effects are treatment, study area, and positive test. So similar to what Mandy was describing, if an individual ever tested positive for one a MOVI test, they were deemed positive just for this analysis. But beta treatment showed a reduced hazard with removal. However, the 95% credible intervals overlap 0, so maybe not statistically significant. And same with our different areas. However, having a positive test ever did increase hazard, which makes complete sense. And our survival estimates with our global model kind of showed that maybe we didn't see increased survival probability, but that could be because of our pretty low numbers, especially after removal. And our cause-specific mortality, I varied it within this global model by herd and treatment. And as you can see, our credible intervals are quite large, so we don't really have any statistically significant evidence that mortality varying by treatment was reduced, or for pneumonia, sorry. And our lamb survival, I just kind of wanted to present just the raw data so that you guys could see what we were working with. In 2018, we collared 23 lambs in the Pine Ridge, and we only collared 5 in the Wildcat Hills. We didn't do any vidding that first year in the Wildcat Hills, and that was just kind of our pilot season to figure out how we were going to work in the Pine Ridge. because that was our main focus, was how we were going to do this treatment in the Pine Ridge. We only had 13% survive, and then a little 40% in the Wildcat Hills. In 2019, this is when we started to conduct our removal. So these were considered our treatment years, 2019 and 2020. 16 were collared in the Pine Ridge, 26 in the Wildcat Hills. Like I said, there were a lot more sheep in the Wildcat Hills to capture. So we caught as many as we could. 31% survived in the Pine Ridge, 54%. All 13 lambs that were available were collared in the Pine Ridge in 2020. And then again, we caught as many as we could in the Wildcat Hills. So still just raw data, what we were looking at. And once started getting into the analysis, again, I'm just presenting our global model. And for our hazard, I looked at how hazard varied by subherd. So between Fort Robinson, Barrel Butte, and Hubbard's Gap, how treatment varied. So treatment consisted of the Wildcat Hills very separately, pre-treatment for the Pine Ridge, and then treatment for Barrel Butte and treatment for Fort Rob. I wanted to break it up so that we could see all of the different treatment aspects. By sex, by birth timing. So birth timing, it was more than 3 days prior to peak lambing, and then more than 3 days after peak lambing, and then by birth weight. So this is kind of a busy slide, but the important ones that I have highlighted in color are the ones that are significant, statistically significant. So we can see that subherd increases hazard for both our Pine Ridge populations. Birth timing, which isn't as important to this workshop, but that was a significant one. So if they were born within 3 days of peak lambing, they saw increased hazard. What was a nice thing to see was that our treatment effect administered in the Barrel Butte was a significant decrease in hazard, as well as the one that makes a lot of sense, the larger birth weight, decreased hazard. So I'll be going into that treatment effect a little bit more for Barrel Butte at least. Also, our lamb survival, you can see that our control group, the mean survival was about 45%. And then for our Fort Robinson herd in the treatment in the Pine Ridge, it really didn't show any improvement. However, our Barrel Butte subherd showed quite an improvement. So it went from 10% to more than 30%. And so we still are seeing Amovi in some of our lambs. So while we successfully removed every chronic carrier you tested from the Pine Ridge, 3 Fort Robinson lambs died this summer of the same strain. And so a potential intermittent carrier also died with the same strain in the fall of 2020. She was negative at February 2019 and 2020 captures, but was positive in February 2018. So what we would consider intermittent, but that's all open to interpretation. And then we did have an additional Fort Robinson ewe that died in late 2021. And she was predated, but she tested positive for Amovi. And she was positive in 2017 and then negative 2018, 2019, and 2020. So it's all going to be very interesting to see how these overlap and correspond with their different strain types. And just to go over everything, in the adults, it showed a decreased hazard and lower probability of mortality due to ammonia, but those results weren't statistically significant. And then in the same vein, our cause-specific mortality analysis, due to the small sample sizes and then varying it by subherd, we didn't have a great power to detect differences in mortality probability, but we did have success in survival of lambs using treatment and varying it by subherd. So Barrel Butte, again, that one on the far eastern side, showed increased lamb survival. That Fort Robinson one, though, right in the middle of our treatment herd in the Pine Ridge, didn't show approval, but that's potentially because we have a very dynamic system up there, and we could have additional reservoirs for Movi, such as that sow belly herd, which is adjacent to it to the west. So we didn't really have a whole lot of information on those. We have tested a few as we can, but there's a lot of landowner disputes out there. So we only get to test those individuals rarely. And also our study didn't look at any rams, but they could easily be, going back and forth between sow belly and Fort Robinson, or there's also always a possibility of domestic sheep interactions. And we do have another grad student at SDSU, Caitlin Bohr, who's investigating more of the domestic sheep pathogen prevalence in Nebraska. So really exciting news to come from her too.
I guess that probably concludes it for Nebraska. A lot of people on the slides to thank here too. And I guess just to quickly summarize a little bit more from Emily, our treatment herds up in the Pine Ridge, Barrel Butte, it did really seem to be pretty effective over in that herd. But as you can see at Fort Robinson, we appear to have some avenues of transmission yet that we do need to try to address. So, but with that, I'll finish up and turn it back over to you, Perry.
Thank you, Todd, and thank you, Emily, for that great presentation. Okay, we are going to try to get back to Jack and the National Bison Range.
So for my presentation, I aimed at hitting as many of the organizers suggested talking points as possible. So I'm going to start with two. First, the organizations and people involved through 2020. And of that group, Amy Lisk was the primary shaker and mover on the managing agency end of things, actually pretty heroic, given all the other things that she had to do, but she kept it moving. Things have changed in 2021. The refuge is transferred to the Confederated Salish and Kootenai tribes. So from here forward, whatever happens with the Bighorn herd will happen under their supervision and management. The guest research team here, I'm not sure why I'm presenting, actually. I think the main reason is that I was the most consistent field presence before the epidemic in question, during, and then after. Second, in terms of motivation for test and remove in this population, I think it's fair to say that the group objective going in included an experimental component. Particularly, that would have been Francis's preference, I think. But in practice, logistics, funding, and population trend dictated something more along the lines of an expedited management action aimed at population recovery. So it's a little bit of a hybrid, but mostly, I think it's fair to say a management action. Also, I should note that results to date are purely descriptive. So this is the study system. Can you see? You can't see that. There we go. So the area in brown is the refuge, the National Bison Range as it existed 2020 and before. And the area in yellow would be the collective annual home range for the Bighorn population. The two are not identical. Our vision of the collective home range for the Bighorn is somewhat different from theirs. And there are two places or two kinds of off-refuge movements. The first is historically short distance explorations of margins of the Bighorn defined home range, and those are typically to the southeast. That's the blue arrow at the southeast corner of the refuge. And then long distance between population ram breeding migrations to and from the West. The regional context is, by the way, this is Francis's slide. Thanks for letting me use it without asking. But this insert to the left shows the regional context. The Bison Range population is an isolate in the regional fabric. But I think that's to some extent true of these other populations that surround it. So the regional context is a relative isolate among several other relative isolates. The habitat, that's the photo in the insert photo, is mountain foothill terrain. So relatively gentle compared to what some of you are used to. This is the population structure, going into test and remove or going into the epidemic. Non-migratory, not hunted, the typical fission-fusion social system with segregation of the sexes outside of rut. No significant population substructure. And then these last two bullets sort of review the last slide. This tenuous connection regionally by male breeding migrations is something that I think it's a tradition or a cultural tradition that was established by a program of genetic rescue in the 1990s. I think that tenuous connection is now severed likely severed by the epidemic. Demography for this population actually goes all the way back to 1922, and I think this is the population that Kevin mentioned as the earliest transplant population he could find. The population counts from 1922 through 1978 are by refuge staff, and they're going to be somewhat approximate. But beginning in 1979, which is our study, they're more or less exact counts from 1979 forward. It's a complicated history demographically and genetically. It includes a genetic and phenotypic rescue, followed by a strong demographic response. So that's the rescue is genetic rescue, phenotypic rescue. Response is roughly the beginning of what we would assume is response to the admixture in the genetic rescue. Population, so ultimately culminating then in a population of 223 resident bighorn in spring of 2016, which is immediately prior to the severe epidemic of fatal pneumonia that is in question here. So in terms of what we knew about the situation in the population before this 2016 epidemic, it's actually fairly extensive. And most of the three clinical assessments listed in this slide, the real-time PCR assay, the C-ELISA assay, and the pathology, the histology, most of that dates from 2015 and 2016, so immediately before the epidemic. And then the other one, which I consider to be a health assessment, are detailed individual life histories for pretty much every individual in the population. No evidence of any negative density dependence in a suite of 8 conditioned influenced traits, typically reproductive traits, but also included in that number would be horn growth rate among rams. Growth rates that were sufficient to produce somewhat over 20 record book rams in the 10 or 15 years leading up to the epidemic. So no evidence of any sort of a health issue and no evidence specifically of MOV prior to spring 2016. That in contrast to the epidemic, in which 90% of 32 fresh carcasses recovered during the epidemic tested positive for MOV. 2 were detected, 2 not detected individuals were symptomatic prior to their death, and 1 indeterminate also symptomatic prior to its death. So collectively, the data suggest, in addition to strain typing of mycoplasma in NBR bighorn and local flocks of domestic sheep, indicate that the cause and source of the 2016 epidemic was a single strain spillover from a local domestic flock in early summer of 2016. So the test and remove part of this demographic story then dates to early 2018, and of course focuses on this small group of roughly 32 survivors of the epidemic. Our methods, capture methods, ground capture by chemical immobilization. Typically, it varied, 1 to 2 crews of 1 to 3 people. Pretty low tech, binocs, backpack, pair of boots, rifle darts and drugs and little more. There were no helicopters, nets, collars, or VITs. And at the very end of the process, during the COVID lockdown, actually just 1 crew of 1 person doing the captures. So on the plus side, at least from my point of view, our approach was pretty surgical with a fairly low population and individual level impact of capture. On the negative side, we lost substantial control of the capture schedule for various reasons, and hence of the preferred experimental design, kind of going in. We used the BAM kits and recommended doses, so no surprise there. This is the capture schedule. All surviving bighorn were captured, sampled, and tested at least once by 5/1/2019. And that's this group over on the left-hand side of the screen. More about the group to the right-hand side over here later. We had a single indeterminate individual, and that individual was later recaptured and tested negative for MOV. And this single shedder, a 6-year-old ewe, was found dead in 2019. So she was effectively removed by predation, we believe. These bars up here at the top, 2018, 2019, and 2020, give the rough timing of lamb drop in this population. So what it means basically is that in 2017 and 2018, we know, or we suspect, based on the testing, that there was at least 1 active shedder present for those lambing and lamb rearing periods. Whereas 2019 was the first lambing period post-epidemic, absent a known shedder. The pattern of lamb survival in the 2 test and remove periods, with and without a known shedder, plus the 4 years preceding the epidemic, looked like this. At the top, this pre-epidemic period, 2012 to 2015, the typical pattern was fairly high early peak or early peak in lamb mortality, primarily due, we think, to predation, followed by very high survival through summer and good recruitment. So this stuff over here on the right, these are basically weaned lambs, fall weaned lambs. When the shedder was present, we had a very different pattern. Many fewer lambs, of course, to test, but there was the same early peak in lamb mortality, followed by this 20 to 70 day period when we started to see symptoms of pneumonia, and we also started to have lamb deaths following symptomatic pneumonia. And then basically no recruitment whatsoever. So a qualitatively different pattern from the pre-epidemic period. Period 3 then is the shedder absent in 2019 and 2020 for the 2019 and 2020 lambing and lamb rearing periods. And the pattern then is again more like the pre-epidemic period. There was the same early peak. Some lambs were weaned, proportionately fewer than in the relative to the early predation. period than in the pre-epidemic period, but certainly different than what we saw when we had a shedder in the population. Let's see, I should say that for the 13 lambs dying in the period to 20 to 70 day window, 9 of those 13 were symptomatic. We observed symptoms of severe pneumonia. 2 of these 9 were collected in terminal condition, both tested positive for MOV DNA. So back to this last cluster of 10 putatively post-clearance captures. This is perhaps the only surviving semi-experimental aspect of the project. These were repeat captures of 5 adult ewes previously captured prior to 5/1/19. The result for these was 3 of 5 were seropositive for MOV, and all 5 again real-time PCR negative for MOV DNA. The group also includes first captures for 5 yearlings recruited in 2019 after the loss of the single shedder ewe. And the result for these is all 5 were negative for both MOV antibody and MOV DNA. So these are both results that Francis and Tom would predict if removal of the single shedder in March of 2019 represented final clearance of MOV from the population. So the organizers asked for a parting thought, and here is mine, and it's based on an N of 1, the single experience with the Bison Range population. But it seems to me, at least barring huge amounts of money and mechanical assistance, that if test and remove is feasible, the population is likely small. And that means they're going to be dealing with generic small population effects. And there's some evidence in our case at NBR of what I would call MOV long haulers. I think it's somewhat similar, or it sounds somewhat similar to what Francis has seen at Asotin. But there are these 2 weights on the back of this population, meaning that successful test and remove is just the first step in recovery. And it may not be, it may be the easiest step. And I think my main regret in terms of my participation in this particular test and remove effort is not advocating at the outset for a follow-up plan of supplementation or other demographic assist for this population, because every year that we go without lamb production, either because MOV is in the population or because of these small population effects or because of these long haulers, it's a deeper hole that we're in and a harder hole to get out of. So I think that's pretty much all I had to offer, and I'll turn it back to you.
Great, thank you, Jack. Okay, if you'll stop sharing your screen, then we can go back to, we've got blocked out a 20-minute Q&A period. So Kezia, I don't know what's popped up in the chat. Don't see any current hands raised.
So we can start from the chat questions if you want, Perry, and then just stop me once you have hands coming up, if that works.
Okay, sounds good.
All right, so the first one, I think we just wanted a quick point of clarification from Francis, but also from others on, so chronic carrier ewes, they seem to die at a higher rate maybe than other ewes in some cases. Should chronic carrier ewes be removed or not? Let's just get that cleared up. Francis, hopefully you're on here. Oh, yes, okay.
Yes, remove the chronic carrier ewes.
Okay. And some of them might naturally remove, but it's still going to benefit you to take the other ones out if you can.
Right. My point was that if you wait a year, you'll have some ewes that turn out, if they test positive, at least in my experience, I'm not saying it's going to be everywhere, but my experience, if you test a year and then retest the next year, some of the ewes won't be positive, and then some of the ewes will have died. So that saves you having to remove them.
Awesome. And then I want to ask a quick one from Pat Nlicka. So are there any external visual signs that might indicate a chronic carrier that could be used in lieu or to accompany testing? And I think that's to all of you who've been doing this. I told him I can't tell, but some of you might have better tricks.
I think sometimes, and I don't know, I like to hear what other people say. And sometimes I think you can, and I think maybe the presence of other issues, perhaps sinus tumors, could contribute to that, but sometimes you can't tell. So it's not totally reliable. Chad, were you gonna chime in?
Yeah, that's a great question. I had the good fortune of following the one shedder for a year and a half, and she looked just like the other animals. I couldn't tell them apart at all. So, but we never found the tumors either, so that might be part of it. But based on my observations, they look exactly like a healthy negative sheep.
And I'll just tag in the countervailing point from Kylie and Helen in BC, where they say many carriers do have some residual pathology, for example, adhesions and small areas of scarring and even active pneumonia. And some have thick nasal discharges. So maybe that's kind of in line with what Francis was saying. Sometimes maybe you can tell, but yeah, maybe it's not reliable. Other things on that point?
I think if they do have nasal sinus tumor, you might be able to catch them with nasal discharge, or even just a ewe that seems to lick her nose more frequently. I think that even if they have a thin nasal discharge that you're not seeing as kind of active snot, they may just have a higher rate of nose licking. I don't know, not tested theory. It's not tested at all. It's just the theory of mine.
And Perry and Cassier, I think a lot of us have wanted the theory of coat molting patterns changing between those that are shedders and those that are clear. And I don't think we ever were able to confirm in Nevada that that was a nice telltale sign. I think we wished, but we could not confirm.
Tom, did you see anything in captivity on the carrier that you could distinguish?
I was gonna pitch in with that exact point. So this U46 that was our chronic carrier for 5 years, you know, the first 3 or 3.5 years, I could never see any difference between her and the other ewes that she was with. But the last year that she was alive, she started losing weight disproportionately to the other ewes in the group, and eventually started showing symptoms, occasional coughing and nasal discharge. So it was, by the end, it was pretty clear that she was ill compared to the others. So I think it just depends on what stage they're in, and you know, that's one animal as an example that she can be indistinguishable from the non-chronic carriers or resistant animals in the same group, even with pretty close observation in pens, and that can go on for years, but then it can also pop up with symptoms. So I think either one's possible.
Okay. Perry, do you have any over there? I've got another big one, but I won't open it unless you. Okay.
Nope, so far we're good.
Okay, so then there were a couple of questions, one from Brent Lahner and one from Shannon Claremont regarding costs. And so Brent's was, what was the mean capture timing and how much was the associated cost? And I mean, that was that was in to Francis, but I think that it would be interesting to hear about that from most of you who've undertaken these projects. And then kind of in that same vein, Shannon asked, given, Frances's project where she's got half of her monitored herds rebounding well and the other half, sort of not necessarily rebounding rapidly, was that the best allocation of funding? Were there other options that could have been taken that might have, paid bigger dividends? And so I think it would be useful to hear Frances first, but then maybe all of the speakers who are willing to speak to issues of cost here.
Yeah, it's a really good question. I should have thought about that before, especially for a management symposium. Of course, it will depend on the situation, what your cost is going to be. But so I guess I don't have an answer for that. We did most of our captures were done by helicopter. And as far as the question, can you repeat Shannon's question again, Cassier?
Yeah, so Shannon was wondering, given that you kind of had this mix of outcomes,
yeah,
is there a better way to have spent that funding?
Yeah, and that's one reason I showed all the other things we tried before we got to test and remove, because we did try every test and remove was kind of our last resort. And I don't know of anything else we could have done that would have been better. The only other choice would be to just depopulate. I think we're going to talk about that later. But what's great about test and remove, and Chad did mention some of the negative effects of trying to re-establish a population from scratch. Oh shoot, forgot the other thing I was going to say. The thing that Jack mentioned, and I've thought of too. I'm not sure about the COVID long haulers, but I feel like we should when the population is really small that you end up with, you might want to be planning on like they did in South Dakota to do a supplement afterwards, translocate some additional sheep in there.
Are other people willing to speak to cost a little bit here?
Yeah, I sure can, Kenzia. So it's highly variable. In Custer State Park, we spent somewhere in the neighborhood of $400,000 to capture, mark, sample each individual twice. So that was very expensive, but the results were really good. Now, contrast that with our Deadwood herd, where we didn't use helicopters. We were able to ground dart everything. In a short amount of time, we probably only spent roughly $30,000, and we had really good results. Now, both of those herds had really small starting numbers, and Jack talked about, the feasibility of this is nice when you have 25 to 30 starting individuals. And I think you can get away with, if you can ground dart, have access to the sheep, you can get away with spending less money and having some pretty good results. So it's highly variable depending on the herd and the area you're in.
Kezia, I'm going to jump in with a question on for hand raised, and Justice Allen has a question. And you want to unmute and go ahead?
I guess one way to get at cost is to know whether, how many collars are being put out, because collars in capturing tend to be the most expensive part of an operation like this. So Frances and others, are you collaring every animal you catch to test, or are you collaring just a portion of a subgroup or population so that you can find that group and then using those as kind of bell mares or to find the other ones?
We're collaring everybody, but we just have some GPS collars. The rest of them are VHF that aren't very expensive. They cost $100 to $200.
And could other projects speak to that as well? Is that general, that people are marking everyone that they catch, and how are folks doing collaring?
In Nebraska, we pretty much collared everything in our smaller herds up in the Pine Ridge. You saw those lower numbers, so we were able to do that. But down on the Wildcat Hills, you know, where we have much larger numbers, it was just a portion of those that we collared. And as far as cost, and maybe answer Brett's question a little bit too from our end, I think we use GPS collars, and I think we were looking at roughly about $1,200 per animal is what we figured. And so, but you know, our general cost overall for the project, I think we're about 200,000, but capture was via helicopter, and for the most part that cost us, you know, $20,000 or a little under, I think, each year.
Just to clarify on that, Todd, that 1,200, is that the collar or collar plus testing?
That was, excuse me, I think I said might have said that wrong there. I was not with the collars. Those were more expensive with the GPS collars, not with the collars.
Gotcha.
Yeah, and one thing to consider is the airtime fees. If you want to do anything that communicates with the satellites, that's also a pretty big cost.
It was I was thinking of VHF collars that we've used in the past, was, you know, cheaper. Obviously the V8 or the GPS are much more expensive. So, and that would have added to it probably. I think those were going about $1,500 with the rate that we got them for. So add that into the mix. So I'd bring it up to probably about, you know, $2,000 to $2,500 per animal when using GPS collars, at least for us anyway.
Well, it's actually a little higher than that, because you got to add the data fees and the testing, and the data fees sometimes are pretty significant.
Perry, do you want to get Will, or do you want me to do another?
Oh, I can get Will. Will, go ahead.
We're just at the beginning of this in the Yakima Canyon area, but we have a herd that's pretty easily seen and is typically in the same portions of their range. So we've attempted to use some rubber belting instead of collars, and you know, we have numbers on them and things like that, so they're highly visual. You can see them pretty easily, and you know, we're trying that out. The cost is negligible as compared to a VHF or GPS collar. We actually had to remove some animals that had VHF collars on them, and we were happy that they had the collars on them, so we could find them easier. But we've tried the belting, and we'll see how that goes.
So there's a question Peach Van Wyck asked for Francis, and this is on a slightly different vein. Any thoughts on what may have maybe contributing to the MOV persistence detected in the lustine? And were you able to strain type any of those animals?
Yeah, I don't want to steal Jenny's thunder, but we'll be talking about that later. And we have strain typed it was, I think Tom mentioned that the ones in captivity had sinus tumors, and which we haven't found anywhere else in Hell's Canyon, and we've looked, and we did strain type the MOV, and it's the same type that's in the rest of Hell's Canyon, or was.
And then another question for you, Francis, from Tony Mong. You mentioned that you feel this is a positive endeavor for large populations. What would be considered a large population? And can you expand on why you think this is worthwhile in bigger populations?
I think it's worthwhile because it worked in a population of 900 sheep. Of course, we were able to have the substructure. If you don't have substructure, it would probably be different. But I just originally, it's definitely easier to do in a small population that's very accessible. But I think that we haven't really seen how far we can go. We've got some projects right now that are in larger populations in the tri-state, and so we'll get more information from them. But I just think it's worthwhile. I'd want to sort of think about all the things that go along with that population, in terms of accessibility and substructure, and whether they have sinus tumors and things like that, or at least what the prevalence, that's the most important thing, is what is the prevalence of MOV and shedding. So yeah, I just think we don't really know. So I think it's worth trying. And I think, we have had some positive results.
And just before we move on, does anyone else want to speak to that issue of potential size constraints? Any of the rest of you who've been engaged in these projects? Okay. Then the last question that I'm aware of is from Brent Lahner, and he asked if any of you experienced any local negative feedback to further removing through test remove animals that were already compromised and in populations that were small, and if so, did that prove to be a significant additional barrier that you had to work through? Was the public bought into this process, or was there pushback?
I have a few words to say on that. So the Rapid City herd in South Dakota is very small, and it's the locals, find their favorite sheep and have a very, for the most part, very loving relationship with them. So there was a time, in this last year that we removed a local, very loved sheep. And through, just kind of talking with the local landowners and kind of educating them that this would be for the better part of the herd, they did understand. But I didn't find that to be a barrier. But by just talking to the locals that were around those areas really helped.
In Nebraska, we didn't really have any issues with people against what we were trying to accomplish in removing some of those sheep. It was probably a greater issue with access being dominated by private land in so much of that area, that just the anti-government sentiment, probably created access issues more than what we were actually trying to do.
Todd, can I just follow up on that for just a second? Can you say just a little bit more about how you overcame those and any constraints that might have been associated with undertaking this on private lands?
Well, we tried to, visit with landowners that had concerns about us being there, but also, I guess, an avenue we've taken is doing a lot of this work through our partners like South Dakota State and so forth, and then letting them take the lead on some of it. Certainly it seemed to help overcome some of those access issues just from that anti-government sentiment. So that's really been helpful for us. But in some cases, we still don't get it, and we just have to work around it. And we work with the helicopter crews, provide them a maps of where they can and can't go. And if the sheep are in those areas, we just don't get them. So we don't get through them all, but we get through a lot of them.
Yeah, I would say it was a great tag team effort. I worked for Game and Parks before going to SDSU, but we knew who liked Todd, and that was a handful of the landowners. And then if there were any that were just against Game and Parks, it was, this is for SDSU, and we're trying to figure out, because usually they didn't like it because they thought Amovi was getting passed to their domestic sheep a lot of the time. So, and then that's why we're doing that survey research with Caitlin Bohr to see what the public's view on Bighorn sheep is and how willing they are to help.
Great. I think that's all the time that we have left for questions. And I think we finished up with all of our questions. So I have, why don't we have a 10-minute break scheduled now? And then we will take up at 6 after the hour.